Hello everyone, and welcome to Think Biochemistry. This channel is dedicated to making biochemistry simple, interesting, and easy to understand for students of pharmacy, medicine, life sciences, biotechnology, nursing, and other health science courses. Our goal is to explain complex biochemical concepts using clear diagrams, real-life examples, animations, and exam-oriented explanations, so that you not only score well in your examinations, but also build a strong conceptual foundation. If you're passionate about understanding life at the molecular level, you've come to the right place. So don't forget to like, subscribe, and turn on the notification bell to stay updated with our latest educational videos. Now, let's begin today's topic. This presentation introduces the fundamental concepts of amino acids, which are the building blocks of proteins. First, it explains the general structure, characteristics, and biological importance of amino acids. Next, it covers the 20 standard amino acids, including their structures, properties, and important biological roles. The presentation then explains the differences between polar and nonpolar amino acids, focusing on their interaction with water and their importance in determining protein structure. Finally, it discusses essential and non-essential amino acids, explaining which amino acids must be obtained from the diet, and which can be synthesized by the body in sufficient amounts. Overall, these topics provide a strong foundation for understanding amino acid properties, protein structure, and biological function. Amino acids are organic compounds that serve as the building blocks of proteins. They are essential for life because proteins perform numerous functions, including structural support, catalysis, transport, signaling, and regulation. A typical amino acid contains an amino group, a carboxyl group, a hydrogen atom, and a variable side chain, known as the R group, attached to a central alpha carbon. The R group determines the unique chemical properties of each amino acid. There are 20 standard proteinogenic amino acids commonly used to synthesize proteins. They are joined together by peptide bonds to form peptides and proteins. The sequence of amino acids determines how a protein folds into its three-dimensional structure and ultimately influences its function. Nutritionally, amino acids are classified as essential, non-essential, and conditionally essential. Essential amino acids must be obtained from the diet because the body cannot synthesize sufficient amounts. Non-essential amino acids can be synthesized by the body, while conditionally essential amino acids may be needed in greater amounts during growth, illness, injury, or physiological stress. Amino acids are important for growth, tissue repair, enzyme activity, hormone and neurotransmitter production, metabolism, and nitrogen balance. Except for glycine, proteinogenic amino acids have a chiral alpha carbon, and ribosomal protein synthesis uses L-amino acids. In summary, amino acids are small molecules that play fundamental roles in protein structure, biological functions, and life. Glycine and alanine. Explanation. Glycine and alanine are two non-essential amino acids, meaning the human body can synthesize them. Both are important components of proteins and participate in several metabolic processes. Glycine is the smallest amino acid, with hydrogen as its side chain, R equals H. Because of its small size and flexibility, glycine allows proteins to adopt tight turns and flexible regions. It is particularly abundant in collagen, where it occurs at every third position and is essential for the collagen triple helix structure. Glycine also participates in the synthesis of heme, purines, creatine, and glutathione, and contributes to neurotransmission in the central nervous system. Its isoelectric point, pi, is approximately 5.97. Alanine has a methyl group as its side chain, R equals CH3, making it a nonpolar hydrophobic amino acid. Alanine is important in energy metabolism, particularly through the glucose-alanine cycle. In this cycle, Muscle transfers nitrogen to the liver in the form of alanine. The liver converts alanine to pyruvate, which can be used for gluconeogenesis, while the nitrogen enters the urea cycle for disposal. Alanine therefore helps connect muscle metabolism, glucose production, and nitrogen metabolism. Its pI is approximately 6.01. In summary, glycine provides flexibility and structural importance to proteins, whereas alanine is especially important in energy and nitrogen metabolism. Cysteine and serine are important proteinogenic amino acids classified as polar, uncharged amino acids. Their side chains contain different functional groups, giving them distinct chemical properties and biological functions. Cysteine contains a sulfhydryl group, SH group, in its side chain. Its structure is HOOC-CHNH2-SH. The reactive SH group allows two cysteine residues to form a disulfide bond, SS, through oxidation. These covalent bonds help stabilize the three-dimensional structure of proteins, especially extracellular proteins such as keratin. Cysteine is also an important component of glutathione, a major cellular antioxidant, and can participate in enzyme catalysis and metal binding. Serine contains a hydroxyl OH group with the structure HOOC-CHNH2-CH2OH. The hydroxyl group can form hydrogen bonds and participate in biochemical reactions. Serine residues are important in the active sites of enzymes and can directly participate in catalysis. They can also undergo phosphorylation, an important mechanism for regulating protein activity and cellular signaling. Serine can be converted to glycine, contributing to one-carbon metabolism. Key difference: cysteine has a reactive SH group, whereas serine has a reactive OH group. Both play important roles in protein structure, enzyme function, and metabolism. Asparagine and aspartic acid are two important proteinogenic, non-essential amino acids involved in protein structure, nitrogen metabolism, and other biochemical pathways. Asparagine, ASN, is a polar, uncharged amino acid. Its side chain contains an amide group, giving it the structure HOOCHNH2-CH2CONH2. Asparagine is important for protein synthesis, folding, and stability. It has a particularly important role in N-linked glycosylation, where carbohydrates are attached to asparagine residues in specific protein sequences. Asparagine also participates in nitrogen metabolism and can serve as a form of nitrogen storage or transport in cells. Aspartic acid, ASP-D, is an acidic amino acid with an additional carboxyl group, COOH, in its side chain. Its structure is HOOC-CHNH2-COOH. At physiological pH, the side chain carboxyl group is predominantly deprotonated and negatively charged. Aspartate donates nitrogen to the urea cycle and participates in nitrogen metabolism. It is also linked to the TCA cycle via oxaloacetate and can act as an excitatory neurotransmitter. Key difference: asparagine has a neutral amide side chain, whereas aspartic acid has an acidic, negatively charged carboxyl side chain. Glutamine and glutamic acid are closely related amino acids that play important roles in nitrogen metabolism, cellular metabolism, and neurotransmission. They differ mainly in the functional group present in their side chains. Glutamine, GLIN, Q, contains an amide group, CONH2, in its side chain, giving it the side chain structure CH2-CH2-CONH2. It is one of the most abundant amino acids in the body and serves as an important carrier of nitrogen between tissues and organs. Glutamine provides nitrogen for the synthesis of nucleotides, amino sugars, and other nitrogen-containing compounds. It also serves as an important metabolic fuel for rapidly dividing cells, particularly intestinal and immune cells. Under conditions such as severe illness, injury, or metabolic stress, glutamine may become conditionally essential. At physiological pH, glutamine exists predominantly as a zwitterion, with an NH3+ alpha amino group and a COO- alpha carboxyl group. Glutamic acid, GLU-E, contains an additional carboxyl group, COOH, in its side chain, giving the side chain CH2CH2COOH. At physiological pH, this side chain carboxyl group is predominantly deprotonated to COO-, so glutamate carries an overall negative charge. Glutamate is the major excitatory neurotransmitter in the central nervous system and is also a precursor of GABA through decarboxylation. Key difference: glutamine has a side chain amide, whereas glutamate has a side chain carboxylate. Proline and tyrosine. Explanation: proline Pro-P and tyrosine Tyr-Y are non-essential amino acids that play important roles in protein structure, cellular signaling, and metabolism. Proline has a unique cyclic structure in which its side chain bonds to the amino nitrogen, forming a 5-membered pyrrolidine ring. This structure restricts rotation of the peptide backbone and therefore reduces its flexibility. Proline is commonly found in turns and bends of proteins and plays an important structural role in collagen. Along with hydroxyproline, it contributes to the stability of the collagen triple helix. Proline is an alpha amino acid containing a secondary amino group. It is sometimes called an amino acid, although this is an older and technically less precise term. Tyrosine is an aromatic amino acid containing a phenyl ring with a para-hydroxyl phenolic OH group. The hydroxyl group can participate in hydrogen bonding and can undergo phosphorylation, making tyrosine residues important in cell signaling and protein regulation. Tyrosine is also a precursor for important molecules such as dopamine, norepinephrine, epinephrine, thyroid hormones, and melanin. At physiological pH 7.4, both amino acids predominantly exist as zwitterions, with a positively charged amino group, NH3+, and a negatively charged carboxylate group, COO-. Proline is especially important for protein structure and backbone flexibility, whereas tyrosine has important roles in protein function, signaling, and metabolism. Arginine and lysine. Explanation: arginine, ARG-R, and lysine, K, are basic amino acids whose side chains are predominantly positively charged at physiological pH. They play important roles in protein structure, molecular interactions, and cellular processes. Arginine has a 3-carbon side chain ending in a guanidino group, CH2,3-NH-CHN-H2. At physiological pH, this group is predominantly present as a positively charged guanidinium ion. Its positive charge is delocalized by resonance, allowing arginine to form strong electrostatic interactions and hydrogen bonds with negatively charged groups. Arginine is important in protein structure, enzyme active sites, and nitric oxide synthesis, where it is converted to nitric oxide and citrulline by nitric oxide synthase. Lysine contains a 4-carbon side chain ending in an epsio amino group, CH2,4-NH2. Its side chain pKa is approximately 10.5, so the epsio amino group is predominantly protonated NH3+ at physiological pH. Lysine participates in salt bridges, hydrogen bonding, and interactions with negatively charged molecules, including DNA and RNA. It also has important roles in protein structure and regulation. At physiological pH, about 7.4, both amino acids have a zwitterionic alpha amino acid backbone with NH3+ and NHCOO- groups, while their basic side chains are predominantly positively charged. Key distinction: arginine has a guanidinium group, whereas lysine has an epsio amino group. Leucine and isoleucine are essential branch chain amino acids, along with valine. Humans cannot synthesize these amino acids, so they must be obtained from dietary sources such as meat, eggs, dairy products, soy, legumes, and nuts. Leucine is a nonpolar, hydrophobic amino acid with a branched aliphatic side chain. It is particularly important for muscle protein synthesis because it activates the mTORC1 signaling pathway, which promotes protein synthesis when amino acid and energy availability are adequate. Leucine can also be oxidized for energy, particularly during prolonged exercise or energy deficiency. Its molecular formula is C6H13NO2, and its pI is approximately 5.98. Isoleucine is also a nonpolar, hydrophobic BCAA with the molecular formula C6H13NO2 and a pI of approximately 6.02. It contributes to muscle metabolism, energy production, and protein metabolism. An important metabolic distinction is that isoleucine is both glucogenic and ketogenic, whereas leucine is strictly ketogenic. BCAAs undergo substantial initial metabolism in skeletal muscle, where they are transaminated before further degradation. Unlike many amino acids, their metabolism is not primarily dependent on the liver. In summary, leucine is particularly important for activating mTORC1 and stimulating muscle protein synthesis, while isoleucine plays important roles in energy metabolism and is both glucogenic and ketogenic. Methionine and threonine. Explanation. Methionine and threonine are two essential amino acids, meaning the human body cannot synthesize them in sufficient amounts and therefore must obtain them from the diet. Methionine, MET-M, is a sulfur-containing nonpolar amino acid. Its side chain contains a thioether group, CH2CH2SCH3. Methionine is important in protein synthesis because AUG is the usual start codon, specifying methionine when translation begins in eukaryotic cells. Methionine is also converted to S-adenosylmethionine, SAM, an important methyl group donor involved in many metabolic reactions. Through the transsulfuration pathway, methionine also contributes to the synthesis of cysteine and other sulfur-containing compounds. Threonine, THR-T, is a polar uncharged amino acid with a hydroxyl-containing side chain, CHOH-CH3. Its hydroxyl group allows threonine to participate in hydrogen bonding and contributes to protein structure and stability. Threonine is particularly important for mucin synthesis, helping maintain the protective gastrointestinal lining. It also contributes to immune function and normal metabolism. Both amino acids are found in meat, fish, eggs, and dairy products. Plant foods such as legumes, nuts, and grains can also provide them, although methionine may be relatively limited in some plant proteins. Key point: methionine is sulfur-containing and nonpolar, whereas threonine is hydroxyl-containing and polar. Explanation: histidine and valine. Histidine and valine are essential amino acids, meaning they cannot be synthesized in sufficient amounts by the human body and therefore must be obtained from the diet. Histidine contains an imidazole ring in its side chain. The imidazole group has a pKa of approximately 6.0, which is close enough to physiological pH to allow histidine to participate in proton transfer and acid-base reactions in proteins. Although the histidine side chain is mostly unprotonated at physiological pH, it can become protonated depending on its local environment. This property makes histidine particularly important in enzyme catalysis and protein function. Histidine residues also play an important role in hemoglobin structure, including coordination with heme iron. In addition, histidine is a precursor for histamine, which functions in immune responses and gastric acid secretion. Dietary sources include meat, fish, eggs, dairy products, and legumes. Valine has a branched, nonpolar hydrophobic side chain, CH-CH3-2. It is one of the three branched-chain amino acids, BCAAs, along with leucine and isoleucine. Valine is required for protein synthesis muscle metabolism, and tissue maintenance. BCAAs can also be oxidized in skeletal muscle and contribute to energy metabolism, particularly during prolonged exercise. Both amino acids are important for protein synthesis, growth, metabolism, and normal tissue function. In structural diagrams, amino acids may be shown in their conventional uncharged form, NH2 and COOH for clarity, although at physiological pH, they predominantly exist as Zwitter ions, NH3+ and COON. Phenylalanine and tryptophan are essential aromatic amino acids, meaning the human body cannot synthesize them in sufficient amounts and must obtain them from the diet. Phenylalanine, PF, has an aromatic benzyl side chain, CH2C6H5. Its phenyl ring is nonpolar and hydrophobic, and contributes to the hydrophobic interactions that help stabilize protein structure. Phenylalanine is converted to tyrosine, which serves as a precursor for dopamine, norepinephrine, epinephrine, and thyroid hormones. Dietary sources include meat, fish, eggs, dairy products, legumes, and nuts. Tryptophan, TRP, contains an indole ring, which is an aromatic heterocyclic structure. The indole nitrogen can participate in hydrogen bonding and contributes to interactions within proteins. Tryptophan is a precursor for serotonin, a neurotransmitter involved in several physiological processes, and melatonin, which helps regulate sleep-wake cycles. Through the kynurenine pathway, tryptophan also contributes to niacin, vitamin B3, synthesis. Both amino acids are important for protein synthesis, growth, metabolism, and normal physiological functions. Explanation: polar and nonpolar amino acids. Amino acids are classified as polar or nonpolar mainly according to the chemical properties of their side chain, R group. The R group is the part of an amino acid that differs from one amino acid to another and determines many of its chemical properties. Polar amino acids are called polar because their side chains have an uneven distribution of electrical charge, or contain functional groups that can interact strongly with water. These groups can form hydrogen bonds or other interactions with water, making polar amino acids generally hydrophilic, or water-loving. They are often found on the surface of proteins, where they can interact with the surrounding aqueous environment. Examples of polar amino acids include serine, threonine, asparagine, glutamine, cysteine, and tyrosine. Amino acids with charged side chains such as aspartate, glutamate, lysine, and arginine are also polar. Nonpolar amino acids have side chains that lack strongly polar or charged groups, and generally do not interact favorably with water. They are therefore described as hydrophobic, meaning water-avoiding. In many proteins, nonpolar amino acids tend to be located in the interior, away from water. Examples include alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. In simple terms, polar amino acids interact favorably with water, whereas nonpolar amino acids tend to avoid water. Explanation: essential and non-essential amino acids. Amino acids are the building blocks of proteins and are essential for growth, tissue repair, metabolism, and many other physiological functions. Based on the body's ability to synthesize them, amino acids are classified as essential, non-essential, and conditionally essential. Essential amino acids cannot be synthesized by the human body in sufficient amounts, and therefore must be obtained from the diet. The 9 essential amino acids in adults are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. They are required for protein synthesis, tissue maintenance, growth, and various metabolic processes. Good dietary sources include meat, fish, eggs, dairy products, legumes, nuts, seeds, and whole grains. Non-essential amino acids can generally be synthesized by the body in sufficient amounts under normal physiological conditions. They include alanine, asparagine, aspartic acid, glutamic acid, glycine, proline, and serine. Some amino acids are conditionally essential, meaning that the body's requirement may exceed its ability to synthesize them under certain conditions. These include arginine, cysteine, glutamine, and tyrosine, particularly during rapid growth, illness, injury, or severe metabolic stress. Therefore, adequate amino acid availability is essential for protein synthesis, growth, tissue repair, energy metabolism, and normal physiological function. A balanced diet containing a variety of protein-rich foods helps meet the body's amino acid requirements.
Thank you for watching. If you enjoyed this video, please give it a like, subscribe to the channel, and click the bell icon to get notified whenever I upload a new video. Your support means a lot and helps the channel grow.